All right, in this lesson we're learning about chromosomes. Here are our syllabus dot points. We are doing topic 3.2. All right, chromosomes are long strands of DNA. They are wrapped around histone proteins and condensed, and you're going to see so many different pictures of how they are depicted. You're going to see plenty of memes in this one as well. All right, prokaryotic chromosomes, and we're talking bacteria here. Remember, there is no nucleus in a prokaryote, so they are just kind of floating around, okay? Floating around in that single-celled organism. They are circular, okay? So they join all the way up. They're not like linear strands of DNA. They do actually connect up. It's quite interesting. They are naked, meaning they do not wrap around proteins at all, and there's only a single copy of each gene because there is that one long piece of DNA. Now, prokaryotes also have things known as plasmids, and I call them bonus DNA because they are extra to that main chromosome. And really interesting features like, for example, antibiotic resistance in bacteria appear on the plasmids. They are not replicated at the same rate, and really interestingly, bacteria can transfer plasmids back and forth to one another. So the characteristics can spread across species. They don't need to be reproducing with them, and they can be released after death. They don't require that new generation to pass on. So quite fascinating, but also quite horrifying when you're talking about something like antibiotic resistance. So they're not actually required for basic functions, those genes on the plasmids, but they are quite beneficial, like we spoke about. All right, Cairns technique. John Cairns used a method to find the length of chromosomes, which was previously very difficult and quite inaccurate to figure out. What he did was he exposed cells to radioactive thymidine, and so they're taken up in the DNA, remembering it's that base T, and then the cells are burst and they are put onto photographic surfaces. So the radioactive thymidine is reacted with a silver compound, and the silver leaves these dark splotches all over the pictures, and they kind of look like this. So the image is developed, and the thymidine-rich areas appear quite dark in the chromosome. So the chromosome can be visualized, and once it's visualized, you can actually try and figure out how long it is. All right, in eukaryotic chromosomes, we know that they are linear pieces of DNA with histone proteins, so it's wrapped around those histone proteins. Different chromosomes carry different genes, all right? Lots and lots of genes in the linear chromosomes there. Now, they are visible during metaphase, remembering that these are all in the nucleus, okay? So we do have nuclei in the eukaryotes. And the sister chromatids, remember we've got that separation there, and it looks quite thick, I suppose. And they replicate during that S phase of interphase. Now, the linear genes means that they can swap during meiosis. When we say linear genes, I mean this gene is always going to be above this one, and it doesn't matter whether it's a single chromatid or a sister chromatid, or it's the other pair or the matching pair. So they're always going to be in that order, and what that means is these legs can actually get tangled, and that means they can swap during meiosis. All right, homologous chromosomes carry the same sequence of genes. They are the matching pair. Now, we might be talking about a chromosome 4, and this one's come from the father, and this one is still a 4, carries all the same genes, but it's come from the mother. So the linear and the sequencing of those linear genes means that I'm going to use the analogy of meals, every single chromosome has the same meals, like it's a 3-course meal, so your entree, your main, and your dessert. But the alleles mean you might actually have a different meal in that course, okay? So this is an allele for, I don't know, how big your ears are, and this one is also the same, but it might be a different version. So it does actually allow for interbreeding within species because every single chromosome that matches in those homologous pairs do have the same linear sequence. So again, we talked about the paternal and the maternal copy, and that's always what's happening here. This is a pair of 2s and this is a pair of 1s, same thing. Now, we have to be able to compare genome size, and this is our delightful BioNinja picture here. We're talking base pairs. When you see BP, we are talking about base pairs. Remember those A, C, Gs, and Ts. So we have to be able to differentiate between genome size and chromosome number. Very different. Humans have approximately 3.2 billion base pairs, and we know we have 46 chromosomes in each cell, but these are all going to be very different across the different species, remembering that some chromosomes can be really, really small and you might have many of them. Some might be just a few long ones. All right, here is another picture. I've still got my scribbles, and you can see there's mostly non-coding DNA versus mostly coding DNA. So if you have a look at bacteria, it means almost all of their almost all of their DNA codes for something important, as opposed to, say, big spaces in between those important genes. All right. Haploid versus diploid. When we are referring to chromosome number, different cells contain different numbers. Now, chromosome number can be haploid or it can be diploid. Haploid means half, right? And diploid means we've got the di for di or double, which means everything's in pairs. So this is a diploid cell, and they're those 46 chromosomes that should appear in that person's cells. And this is the haploid chromosomal number there. And that is, say, this one's got an X and a Y, that's going to make it a male. So in this particular one, if we split it, it's going to be a sperm cell. So it only appears in the gametes, those sex cells, in our case, the sperm and the egg. Diploid cells are also written as 2N, the 2 indicating that there are 2 of each pair. So they are formed by fertilization when the egg meets the sperm, or during mitosis when a cell just replicates itself. All right, it's the full set. It's got everything it needs. Pairs can allow that one gene be silent, which means if you've got one from mum and you've got one from dad, if this particular one has a gene for, whether your earlobes attach or something, and it's a bit dodgy, then your body will actually use this one and code for this one. All right, haploid is written as N, meaning that it's only got one of each pair. So it's the half set, one of each of those homologous pairs. It's formed during meiosis, right? And they are only found in gametes, the sex cells. Interestingly, some organisms actually have haploid cells as part of their main cell type in part of their life cycle, but that is not us. All right, to compare chromosome numbers, let's have a look. We've got some really a roundworm parasite, not very exciting. Only got 4 chromosomes in each cell. The Oryza sativa is a rice plant. Homo sapiens, we know, are humans with 46. The pan-troglodytes are the chimps, and the Canis familiaris, which are our puppies, have 78 in our diploid chromosome there. So chimps and humans are a great example of the fact that a change can happen during evolution of a species, and that might actually give a rise to a difference in chromosome number. So the H here stands for human, we've got chimp, we've got gorilla, and we've got orangutan. So if we actually have a look at these, this is the chromosome 7, and they're quite similar, right? Really, really similar genes on these chromosomes, but over time things have started to drift. The chromosome number dictates the outcomes when we are cross-species breeding. Now, a horse has 64 as its diploid number. A donkey has 62. So when it splits and makes sex cells and then we add them together, we get a donkey where it's, a mule where its diploid number is 63, and that causes problems because in order for us to halve a 63 to get some sex cells, it's not really going to work so well. All right, karyorgrams are images which show ordered paired chromosomes, and the karyotype is what we look at when we actually try and find to see if everything is in its right place. And we can stain the chromosomes, and what happens is bands arise and they look like stripy socks, so we can find if there are abnormalities. They used to be processed with a camera, film, and scissors and glue. It is now done with a camera, a slide scanner, and a computer. So chromosomes, if we look at it like this, we can determine whether a patient is a male or a female based on their sex chromosomes. Every other kind of chromosome is called an autosome. So all these ones here, 1 to 22, they're autosomes, but the XY are our sex chromosomes. Chromosome abnormalities come in so many different forms, and you can have big chunks duplicated, you can have chunks inverted, so twisted upside down, you can have chunks deleted, chunks inserted, and also translocated, which means they do a bit of a switcheroo. Okay, so many different things that could potentially happen, and we'll learn more about that when we do meiosis. Here are our syllabus dot points. Please make sure you have a read.